You know, ever since humans first started farming, we've had one single goal: make our crops better. We're always looking for more food, tastier food, and plants that are just plain tougher. So, how did we go from just hoping for lucky breaks to actually taking direct control of a plant's DNA? Well, let's get into the fascinating science of plant mutagenesis.
Okay, let's start with a number. A really, really big number. 9.7 billion. That's how many people we're expecting to have on this planet by the year 2050. And that, well, that's a huge challenge for all of us.
Which brings us to the big question, the one that drives all of this work. How on earth are we going to feed everyone? I mean, we've got limited land, limited water, and a climate that keeps throwing us curveballs. So what's the plan? Well, a big part of the answer is pretty wild. We have to hack evolution itself.
Think about it. For hundreds of years, plant breeding was basically a waiting game. You find a plant you like, maybe it's a little bigger, a little tastier, and you breed it. But you're just waiting for nature to give you a lucky break, right? A random genetic oopsie, a mutation. So scientists thought, what if we don't have to wait? And they built a toolkit to speed the whole thing up. It's called mutation breeding.
And let me tell you, this wasn't some small thing. This was a game changer. Intentionally causing mutations, what scientists call induced mutagenesis, became one of the most important tools for improving our crops. Suddenly, instead of waiting for lucky accidents, breeders could create this huge library of genetic options to choose from.
So this original toolkit basically had two main ways to get the job done. You had physical methods like radiation, and you had chemical methods. Both are super powerful, but they're not exactly subtle. Think of it like a shotgun approach. You're just blasting the plant's DNA with a ton of changes and hoping that something good comes out of it. All right, so let's dig into that physical method. We're talking about zapping plants with stuff like high-energy gamma rays. These rays just punch right through the plant's tissues, acting like billions of tiny little bullets. They cause some serious changes to the DNA, breaking it, mixing it up. I mean, it's a pretty blunt tool for sure, but man, does it create a ton of genetic variety and fast.
Okay, I know this chart looks a little intense, but it shows that this isn't just random chaos. There's a method to the madness. You start by picking your plant, right? Then you hit it with that gamma radiation. But here's where the real work kicks in. You have to grow literally thousands of these mutated plants and screen every single one to see if you got anything good. After years, and I mean years of testing, you might finally have a new, better variety ready for the farm.
Now, the chemical method is a little different. It's more subtle, but it's still pretty random. The go-to chemical for this is something called EMS. What it does is it causes point mutations. Basically, it changes a single letter in the plant's DNA code. It's not the massive damage you see with radiation. It's more like making millions of tiny typos. But those typos create a whole new world of genetic possibilities to explore.
And was this chemical technique a success? Oh yeah, big time. You've probably got the results in your pantry right now. Seriously, the soybeans for your tofu, the wheat for your bread, the rice that feeds, billions of people, a lot of these staple crops we depend on were improved using this very method. It's foundational to our food supply.
So for a long time, this shotgun method was the state of the art. And it worked. It gave us literally thousands of better crop varieties. But you know how science is, it never stands still. The next big step forward was going to be a massive leap. A true revolution. We're talking about going from random chance to surgical precision.
You see, the big drawback with the shotgun is that it's messy. For every one good mutation you get, you might get hundreds of useless ones or even bad ones. And then you have to spend generations breeding those out. It's just slow and inefficient. So scientists started asking a really important question. What if we could just skip all that? What if we could edit just the one single gene we want to change? And the answer to that question: CRISPR/Cas9. It is absolutely revolutionary. The easiest way to think about it is like a find and replace tool for DNA. It's got two key parts. First, there's a guide RNA. Think of it like GPS coordinates that tell the tool exactly where to go in the massive genome. Then, there's the Cas9 enzyme, which is basically a pair of molecular scissors that makes a perfect clean cut. Suddenly, we could edit the code of life like we edit a document. This really just spells out the difference perfectly. We went from the shotgun just spraying random changes all over the place to a sniper rifle. With CRISPR, you can take aim at one specific gene, make one specific, predictable change, and get the result you want in way, way less time. It's just a whole new level of control.
Okay, so this is all cool science, but what does it actually do for our food? I mean, what are the results? Well, this isn't just something happening in a lab anymore. These techniques are producing real, tangible benefits out in the field right now.
Let's look at rapeseed. That's where we get canola oil. You can see right here on the chart, a normal plant, the wild type, has about 43% oil content. Pretty good. But by using gene editing to tweak a couple of specific genes, scientists created new versions that got up to 47% and even 50% oil content. That is a huge jump. We're talking more oil from the same amount of crop, which is a big deal both economically and for nutrition.
But hey, let's give some credit to the old school methods too, because they created some legendary crops. You've got diamond barley, which was shorter and gave a better yield, and it totally revolutionized brewing in Europe. Then there's golden promise barley, still a go-to for amazing beer and whiskey. And we can't forget Calrose 76 rice. It was a semi-dwarf plant that wouldn't fall over in the wind, a problem called lodging, and that made it a crucial part of the Green Revolution, helping to prevent famine.
So when you put it all together, what can we actually do with this amazing toolkit? Well, the list is pretty incredible. We can engineer plants for better yield. We can give them built-in resistance to diseases and pests. We can pack them with more nutrients. We can make them tough enough to handle drought, heat, or salty soil. We can even make them last longer, which helps cut down on food waste. The possibilities are just enormous.
Of course, you know the old saying, with great power comes great responsibility. And that's absolutely true here. So that brings up a really important point. How do we make sure this powerful technology is used safely?
Well, before any of these new crops ever get close to a dinner plate, they have to pass a whole series of tough safety tests. As you can see here, it's a multi-step process. First, scientists have to know exactly what genetic change was made. Then they check for any possible health issues. Is it toxic? Could it cause allergies? After that, they look at the environmental impact. And even when a crop gets the green light, it's still monitored for any unexpected long-term effects. So, here we are. We've gone from just watching evolution happen to actually taking part in it. We can now edit the fundamental building blocks of our food. The science has handed us these incredible tools. The big question for us, for society, is what do we build next? How do we use this power wisely to create the future we want?